We have just earlier in the day heard about motor proteins, cargo transport in the context of the narrow axons from Sandhya. And we have looked at several kinds of motor proteins, how they may attach to synaptic vesicles, and the role of the cytoskeleton, which or the other structures which are present in these narrow path, and how that might control or stop actually these cargoes from moving. So my lab is also primarily interested in transport and motor proteins, but we have increasingly over the years, because of various reasons, also moved towards understanding this partnership between motor proteins and the membranes on which mostly all the time these motors live their lives. And that's what I'm going to talk to you about. But although we started from a very biophysical point of view in looking at motors, we have also gradually moved and perhaps paid more attention these days to understand physiological processes, how this partnership between motor proteins and the membranes that they sit on, how that can affect you and me in the sense of, at an animal level. And that's what I say when I mean physiology. And I will try to address this to the best of my ability in the context of, two biological problems. One is essentially how much fat is floating around in your blood, okay? And that, to any layman, they would understand that that is important to understand because the amount of fat in that deposits in various kinds of cells in your bodies and can be very detrimental to your health. And the other, which maybe I will come to later, is from a more biophysical point of view, try to understand how this, the motors and the membranes, largely motors, how they might affect the degradation of pathogens in some context. I will not really say pathogens, but degradation of particles inside immune cells. So, I can do this, right. So very quickly, microtubule motors, we know about them now broadly, and here is a very fairly dramatic representation of what microtubule motors can do, where you are seeing here, a cell type which, is under the skin of certain animals, like let's say chameleons, which can change color. And this is kind of the periphery of the cell. And even without the video not being run, you realize that there must be radial structures inside this cell. And that's why you can see this kind of black, radial pattern here. And when I run this, maybe it'll be a little clearer. You can see that upon stimulation by a particular hormone, the names are not important right now, you can see that, there are a lot of these black structures which went towards the periphery, and by some other treatment, you can get them to go the other way. And these are actually little balls of ink, pigment granules. And when the chameleon hiding in the bushes sees a cat, it might do something like this to change its color, okay? Or the other way. And this transport, which you could see happening here, takes about maybe 10 minutes, 5 to 10 minutes of that order. And it happens along microtubules, and it is caused by these motors. And one of the things that one can do, and this is a very, very simplistic picture, where here is a motor which is tied to one of these pigment balls, and it tries to move along microtubules in this manner. And you can do various kinds of assays. One of them is shown here, where you can try to measure how strong this motor is by shining a laser light like this through this, and it focuses on this little cargo that it is carrying, and it can then prevent the motor from walking further, and the motor can fall back in the trap. And maybe we'll come to those things later, tomorrow or day after. So this is a very physicist's way of looking at the problem, and this is a fine question to ask, because that's where, coming down to the nuts and bolts of how these motor proteins work. And many people, including ourselves, have addressed these sort of questions. But beyond that, as Sandhya also pointed out, if this is a typical vesicle inside, some cell type in your body, in this specific case, it's a synaptic vesicle, you will see a membrane around this vesicle, a lipid membrane shown here in green, decorated by gazillions of proteins like this, and which have their own functions, and a few of these proteins, let's say this purple one here, could be a motor, and a few more. Okay, so in this situation, how to understand the net function of this object, and then further on to try to relate it with something bigger that is happening in biology is certainly a tough ask. Now, one of the things that when you try to address something like this, you try to find your niche area, something that you can do and contribute meaningfully. So we have been very interested, more out of circumstance than anything, that in this attachment between these motor, of course so this is the membrane covering, let's say, a synaptic vesicle or an endosome or any generic kind of cargo inside the cells of your body. And inside it is something which will not concern us right now, but this is a double layer membrane, as you can clearly see in this, and this is what we are all used to, okay? And to this membrane, by various proteins or various mechanisms, we will not go into those details right now, there are motor proteins attached. Of course, there are many other proteins which we are not showing here in this picture. And it is important for us, I mean, we all realize is that the attachment of the motor to this structure could either be directly, a part of the motor binds to this lipid directly, that is one possibility, or the motor attaches to some other protein, let's say call it protein X, which in turn attaches to this lipid. So such complicated structures can happen, and there are many, many examples in the literature. And not only the protein part here is interesting in the sense that the motor is of course a protein or the intermediate protein, but the lipids themselves also, and their geometrical structure within this membrane, these are all important considerations, particularly the aspect of what the lipids mean to this organization of motors is something which has been very not addressed very often, and that's something which we will come to later. Now, however, this is not the only kind of cargo that or vesicle that exists inside cells. There is a very important kind of cargo which is fundamentally different from this, and that looks like this. Okay, so these are fat bodies inside practically every kind of cell in your body, and what is different here is that the core of the particle is a hydrophobic compartment. Okay, it carries fat, so therefore the membrane surrounding it cannot be a bilayer in the sense that you had seen earlier. Okay, because in this situation you had the polar groups of the phospholipid facing inside, but now if you fill it with fat, you can't do that. So you remove the inner layer, and so you have only the outer layer present. So these kind of fatty cargoes, so they contain triacyl glycerols, for example, a glycerol conjugated to 3 fatty acid carbon chains, and esterified cholesterol, and other fats of that ilk, which constitute essentially a neutral core. Okay. Now the important point to note is that once you have a monolayer membrane surrounding this particle like this, then the rules of these motors binding to this are completely going to be different. Okay, because something that can bind to a bilayer may not bind to a monolayer, and vice versa. But why is this important to biology? Okay, and why is it important to understand these connections, and why is it important to possibly to health and therapeutics is something that we have been very interested in. Now, these particles here, they are called lipid droplets because you can see that they are the shape of drops. So they are droplets and they are filled with lipid, in this case neutral lipids, so they are called lipid droplets. Okay, and lipid droplets are essentially why your car works. Okay, because the fuel that you use is these in the lipid droplets in all the critters that died 1 million years ago. All the fuel is coming from the lipid droplets which were stored in those organisms. Okay, so lipid droplets and production of lipid droplets, let's say in algae or in microorganisms, is extremely important for the biofuel industry as well, but that's not something that we will get into right now. Okay. So here is a movie where what we have done is that there are particular fluorescent dyes which will enter the neutral lipid, and when they enter the core of this lipid, basically in this area, that dye, if you give the dye from outside, the chemical environment is different, and then this dye starts to fluoresce. Okay, it will not fluoresce here outside, it will fluoresce only inside. How exactly, let's not get into that right now. But by doing so, if you just the dye itself is hydrophobic. Okay, so it can easily go through the plasma membrane of the cell. Okay, and now it starts to stain these lipid droplets, and you can see that these, this movie was over a period of a few minutes. I don't remember exactly what, but you can see that these lipid droplets are extremely dynamic and they move around. And this motion of lipid droplets has been realized over the last 5, 10, 15 years or so. And it is extremely important now, this motion, because what are these lipid droplets? They are small packets of energy, of fat, okay? And when these lipid droplets move around and interact with other organelles inside the cell, they can exchange energy, lipids, in the form of lipids and various other proteins, okay? And they are a very important part of your metabolism because this is the most concentrated source of energy in your body, okay? Why? Because these lipids with long fatty acid chains, hydrocarbon chains, because they are neutral, you can pack a lot of them in a small volume. If they were charged, you could not have done that, right? So that's why, to understand how these lipid droplets interact with other cellular, intracellular organs is becoming very, very important, and they practically interact with every kind of organelle inside the cell that you can think of. One example, for example, is that they interact with mitochondria. Okay, they interact with mitochondria, particularly when you don't eat. Okay, when there is a lack of glucose, when you don't have glucose to move around your hands and all, then you now switch to using fat, the energy in that inside this, and that can only be derived when these lipid droplets go and interact with the mitochondria, and this fat is, by various mechanisms which we won't go into, taken out and eventually converted into adenosine triphosphate, okay, ATP, to allow you to move and all that. Okay, right. So, what was the way we kind of got into this all this business is that what Pradeep, a student in my lab about 7 or 8 years ago, maybe a little bit more, what Pradeep did is that he purified, he was trying to do something else, but serendipitously we purified these kind of lipid droplets from the liver of a rat, okay? And when we took these lipid droplets, and here this is not inside the cell, this is in vitro, this is happening on a cover slip. We stuck a microtubule on this cover slip like this. Is a microtubule. It is stuck to this cover slip. You won't see it because it is in low contrast. And then when you flow in these lipid droplets that you have purified from the liver, you see that very frequently these lipid droplets will come and attach to the microtubule, move over long distances, and eventually they will detach, or sometimes they just go on until the microtubule ends. And this was completely crazy because every last lipid droplet we saw was moving like this. I mean, this is rare to see in this kind of in vitro assays. What we could also do is that, again, as I had told you, we could catch this lipid droplet in a laser beam and put it here and engineer these events. And we saw that, look, this is a very frequently occurring phenomena. And remember, this will be important as we go along, that these lipid droplets were purified from the liver of a rat which was feeding, okay, which was a fed rat. Why do I use the word fed and fasted? Because earlier I alluded to metabolism and use of energy, okay. So therefore this fed versus fasted is going to be important. And what I'm not going to show you here is that when we did the same experiment with A rat which had been fasted overnight, you absolutely don't see this motion. Okay, so or very little of it, and this completely changes, which kind of suggested to us, although in a way this was a very, I mean, we had to do a lot of work, and this is still under progress, that this motion may have something to do with the metabolic state of your body and with the utilization of what is inside this. Okay, so we went on, in various contexts, but before that, I want to show you this particular picture. When we saw this happening, we thought, okay, let us look at sections of the liver, okay, and what does this lipid droplet look like inside the liver? And this is a thin section. So the way, perhaps some of you are not familiar with these experiments, what you can do is that you can cut out the tissue from, you have to perfuse the tissue to kind of, wash out the blood inside the liver, and you cut very thin sections, 5, 10 of that order, micron sections of the tissue, and then you place it on a cover slip, and you now you can perfuse this also with the same dye which I had shown you earlier, the one which went inside the cells, and it marked the lipid droplets. And you can see that in this section, these red outlines are individual cells of the liver. These are called hepatocytes, okay? And we were seeing from Ramre's talk how these hepatocytes are arranged in these beautiful patterns inside the liver. I'm showing a much more zoomed in view, so you won't see that pattern, but these are hepatocytes, and these are the boundaries of these hepatocytes. And here you see these, these little balls of fat. These are the lipid droplets inside the hepatocytes, okay? When I do the same experiment on a fasted rat, this is what things look like, okay? And which is immediately telling you that there is much more fat inside the liver, your liver or the rat's liver, when you are in a fasted state, okay? But what is the importance of this, okay? Why is this important? Because these balls of fat here are going to get chewed up by various mechanisms which we'll not get into right now, and they're going to get repackaged into another kind of smaller lipid droplet, which is called very low density lipoprotein. And many of you would be familiar with very low density lipoprotein. This is what makes up a lot of the serum triglycerides, basically the fat in your blood, okay? And too much of this in your blood report is bad news, okay? So these particles, these blobs here, are recycled and reused to make these very low density lipoprotein particles and secreted out into blood. They will then go into all kinds of tissues through the blood, and because they are so small, 40, 50 nanometer of that order, they can get into the narrow capillaries, okay, and provide energy and lipids to all kinds of cells in your body. But now the question is that if you can clearly see that there are very few lipid droplets here, and there are many more lipid droplets here. And there was a certain mechanism which, let's say, has a certain rate constant, by which you take out a part of these shiny things and secrete them out into blood. If you continue to do that in the fasted state here, then you would be in serious trouble, okay? Because your serum triglycerides could go up about 5, and it's something, in fact, which we showed. I'm not going to go into this right now. The amount of fat in your liver, your serum triglycerides, would fluctuate wildly whenever you stop eating, right? You don't want that to happen, because if you had a huge amount of triglyceride being secreted out, then that could, in principle, and again, I'm not going to show you the details, go into all kinds of tissues in your body and create havoc, okay? And so this is what I'm showing you here. This is in your blood, essentially, okay? This is the amount of fat in your blood. You can see that between the fed and the fasted state, there is no change. So this is a thin layer chromatograph. Don't worry about the mechanism. This band intensity shows you the amount of fat in your blood, okay? Let us take it like that. You can see that between the fed and the fasted state, there is hardly any change. Whereas inside the liver, there is a fasted state, there is much more fat here, okay? And which you can see here also. So there must be some mechanism to block the flow or reduce the flow of fat from the liver into the blood in the fasted state. every time when you fast, okay? And this is something which had, I mean, at least to the best of our knowledge, never been discussed, never been shown, okay? So we so this is something I saw Houston, we have a problem that if you have this kind of a transition happening, and unless a mechanism exists, which is until that date when we did was unknown, I mean, how do you deal with this, okay? So what we figured out, again, I'm not going to go into the details of this because it's published work. You can take a look at this. Okay, I should not go so fast because I have to. Yeah, ask question, please. It's not fragmentation really. Why there are just many more of the lipid droplets. We don't see any of. Yeah, that's to the best of our ability. That's what we can see. Anything else? Yeah, please.
That we are observing no motion of the lipid droplets in the fasted fat liver. Is it due to lack of this ATP?
No. ATP is there. I mean, lack of ATP would be another problem. You don't want lack of ATP in the fasted state because if you had, it's not that the ATP levels also change so much in the fasted state because then your bodily functions would stop. I'm going to come to that. I'm going to come to that. Yes, please.
So when you talk about those devices carrying the neutral lipid, so you showed that when you check for the fed, there was a motion, but when you check for the fasted rats, you did not see a motion. So could it be because of the receptors, different receptors?
That's what I'm going. So by over the next 5 or 6 years, we did a lot of things. I mean, some we did in cultured cells, some we did in animals, some we did reconstitution assays. But eventually, this whole thing came down to a pathway where the connection between a motor protein and the lipid membrane on that lipid droplet is changing. I will tell you what that is, but this change is happening under the control of insulin. Okay, and that makes, to me, it's really exciting because that immediately kind of explains the response to fed and between the fed and fasted. Okay, the difference. Okay, because you know that in your fed state, when you have eaten and that you will secrete insulin, and there's going to be more insulin in your liver and other organs also. Now, before I go to that, one thing I forgot to tell you is, so there is much more fat in the liver here and in the fasted state and less in the fed state. Where is this fat coming from? It is coming from your adipose tissue. Okay, it is coming from your fat reserves in your body, because if you have not eaten, there are hormones which now signal, look boss, there is no glucose around, so let us chew up the fat in the adipose tissue, and that fat primarily goes to the liver first. Okay, and the liver, has being the metabolic center of the body, is essentially, you can think of it as a postman who will now redistribute this fat to all the other parts of your body by secreting VLDL. Okay, so this fat, more in the fasted state, is adipose tissue derived fat. Okay, it is coming from there, all right, and it is accumulating here. So you can imagine in the fasted state, your adipose tissue will have less fat, right? Because you're using it up, and that's why when mountaineers go, before they go, on long treks, they eat a lot and they get fat actually. Okay, and when they come down, they are really thin. Okay, so the reverse is happening there because you're using up the fat in your adipose tissue. Yes. So that is the control mechanism I'm coming to now. That's what I've not come to yet. Yes.
I have a related question.
Yes, yeah.
Very stupid question. Okay. So normally when we go for ultrasound, when you are having some liver disease, or,
Right, right.
You're very fat, doctor normally recommends ultrasound, right?
Correct.
And most of the time they say that there is fatty infiltration of liver, right? And most of the time you go for the scan with empty stomach, right?
Right.
Does that mean, whatever we get?
You're talking about the liver, scan of the liver, yes.
Yeah.
Yeah.
So those, because now we know that when we are fasted, right, there is large accumulation of the fat in the liver, right? So is it possible?
So I don't, how long is that fasting that you are
about 8
referring?
hours.
Yeah, so it's similar to that. See, these experiments are done in rats. Now, I don't, so this is a 12-hour fasting in rats. I don't know what is the equivalent hours in humans. Okay, so I would hesitate to give a direct answer to that.
It would be interesting.
Because I can't do this experiment in humans, right? But I'm fairly sure this is what is happening at some point of time. Yeah. Okay. So yeah. So what? So again, just to repeat the question, that a certain amount of fat is being secreted out. So this is a single cell of the liver. Okay, it is a hepatocyte. Okay, it's called a hepatocyte. It is a single cell inside your liver, and it is joined to another cell, and so on here. What we figured out, this is a cartoon, so don't take it very seriously, but more or less what is happening is that you have microtubules arranged like this in these hepatocytes. Okay, they have a certain polarity, and when in the fed state your insulin levels are high, insulin activates a protein called ARF1. The names are not important. It's a GTPase. Okay, it can hydrolyze GTP, and this ARF1 has this little lipid tail hanging out from one end, which it can insert into this membrane. Okay, and it can go and sit on this surface. Now, the interesting part is that once this ARF1 goes and sits on the membrane of a lipid droplet like this, imagine this thing is going and sitting here, okay? This is a spherical, I don't have anything spherical here, but it goes and sits here, okay? There, it hydrolyzes the GTP and changes its conformation, okay? Because, when you hydrolyze GTP, let's say a phosphate group is, leaving somewhere, so there is a change in net electrical charge. And so the protein will like to be, if it was like this, maybe it likes to be like this after that, okay? So this protein now sits on the membrane, remember that phospholipid monolayer membrane, and undergoes some protein conformational change. And when it does that, it pinches out small lipid droplets from the large lipid droplet, okay? Because it has attached and it is stuck inside and it goes like this, and this has been shown by others, okay? This is not something that we have shown, this has been shown by. But the effect of that is that the large, let's call it the mother lipid droplet, okay? The mother lipid droplet had a certain amount of phospholipids on the surface and a certain amount of neutral lipids inside. So there was a certain surface to volume ratio, because of which it had a certain coverage of phospholipids on the surface. Now remember that inside is fat, hydrophobic, outside is water. So if I now start removing phospholipids from the surface, this surface tension will go up like crazy, right? Because this inside fat doesn't like the water outside, okay? So this process of ARF1 sitting on those lipid droplets and pinching off small lipid droplets removes more surface compared to volume, because it's a surface phenomenon, right? And when that happens, these resultant lipid droplets, the mother lipid droplet with something pinched off, whatever you get, is much more reactive, right? Because it has less clothes to cover it. It will want to go and fuse with some other membrane so that it can acquire phospholipids, okay? It's as simple as that, okay? And this has been known. So what you do by the action of insulin is that one of the things that you do is that you make the surface of the lipid droplet more reactive, means you increase the surface tension, or in other words, you make this lipid droplet prone to now, it is looking for somebody to fuse with, okay, so that it can get phospholipids. But the point is that you don't want this lipid droplet to go and fuse with the next membrane right next to it, okay, because the purpose of one purpose of the lipid droplet in the liver is to supply this yellow stuff for making this yellow stuff, right? So you want this lipid droplet which has this reactive membrane to fuse with a specific organelle inside the cell where these VLDL particles are made, but that organelle is the smooth endoplasmic reticulum. Okay, you know that in cells that you have rough and smooth endoplasmic reticulum. The rough endoplasmic reticulum is rough because it has lots of ribosomes on it, so in the electron microscope it looks rough. The smooth endoplasmic reticulum is does not have so. I mean, this division is not very clear, but still, I mean, the smooth endoplasmic reticulum has less ribosomes, and very interestingly, the smooth endoplasmic reticulum is present largely at the periphery of these hepatocytes. Okay, I don't have I could have shown you that figure, I forgot that. Okay, you can see that the smooth endoplasmic reticulum, that's what we have shown here in the way, it is nicely at the periphery of these hepatocytes. Okay, it's not there everywhere. So now we have a problem that here is a reactive lipid droplet wanting to give its lipid to this guy who's not there, right? So this is probably a step in the control regulation. What we figured out is that ARF1 has two jobs, okay? One, it makes this surface red, reactive. The second thing it does is that it also recruits another protein called phospholipase D1, okay? Phospholipase D, what does that mean to people who are not familiar with is that phospholipase D is an enzyme which will go and chop off something from a phospholipid, okay? So what it does is that it chops off a part of the phosphatidylcholine on the surface of these lipid droplets and converts that phosphatidylcholine to a simpler phospholipid called phosphatidic acid. Okay? Phosphatidic acid, that means it must have a negative charge, okay, because it has given off its protons at pH approximately 7, so it has a negative charge, okay. So phosphatidic acid has two important properties. First, it has a negative charge. We will come to that in a minute. Second, while phosphatidylcholine is like this in the membrane, phosphatidic acid is like this in the membrane. Okay, so the head is smaller than the display of the legs, so it induces curvature in the membrane by this property. Okay, if you had only cylindrical stuff, like this, then you would make a flat membrane. But if you have various lipids of different, curvatures, then you can bend the membrane. There are other things which also bend the membrane, but this is one of them. Now, phosphatidic acid, remember first what I said, because it is an acid, it will, release protons and it will become negatively charged. What happens is that the kinesin 1 motor, there are several kinesins, this is the conventional kinesin called kinesin 1. The kinesin 1 motor comes along and attaches to this phosphatidic acid directly, okay. We don't have any evidence for any intermediate protein. I will come to that in a little bit, okay. It comes and attaches to this phosphatidic acid directly, and when that happens, you have the machine which can transport this to the end of this microtubule, and that is exactly what we were seeing when we saw this. Okay, this is going towards the plus end of the microtubule. This is not inside the cell anymore, but we know the polarity, okay. So these lipid droplets are being, I'm not going to go into details, are being carried by kinesin towards you can imagine towards the periphery of the hepatocyte, okay, although this is not inside the cell, okay. But why do you need all this, okay? Now suppose in the fasted state, you won't have insulin here, right? This entire pathway will be toned down or, reduced. So now you have many more lipid droplets in the fasted state, remember, okay? But the reduction in this pathway allows you to secrete the same amount of VLDL. Right? Because your internal content has gone up 30-fold, 20-fold, but you're still continuing to secrete the same amount of VLDL because this activity of taking the lipid droplet to the smooth endoplasmic reticulum has gone down. That's one of the mechanisms. There may be other mechanisms also, okay? So perhaps this is one mechanism by which you control the outflow of fat from the liver across metabolic states, okay? And so imagine if you were to take, so you never take insulin in the fasted state, patients of diabetes, okay? You always take insulin after you eat. What we did is that we took rats I mean, you can do this with rats again. You take a rat, you take it to a fasted state and give it insulin, okay? You're not supposed to do that. You give it insulin to test this. You can cut open the rat and you check, you see there's massive accumulation of fat in the heart tissue, okay? We have seen that and it's published work. So this, you now understand that if you short circuit this pathway, when you are forcing insulin in, when there is still so much lipid droplet inside the liver, it can be pretty bad for you, okay? So this, so now the question is that, look, if this mechanism, yes, Sandhya.
Why is the liver more sensitive than the heart? Clearly, right? Because in just fasted animals, we see this increase. I've never seen a report.
Because perhaps this mechanism was needed only in the liver because the adipose fat comes to the liver first. It doesn't go to, you said somewhere else. Compared to, you said?
Compared to the heart.
Compared to the heart. It doesn't go directly to the other tissues. Almost all the fat that is coming from adipose stores has to go to the liver first.
Does that mean that the insulin treatment that you give in fasted animals, is that response only in the heart or all tissues under?
No, it's influencing the liver. It's not.
It's through the liver.
Yeah, it's through the liver. Because it's clearing out this fat from the liver and it's going to the heart. It's short circuiting this pathway. Yes, because this control is lost.
So the next question I had is, what is the specificity of interaction with the smooth ER? Why can't it pick up other kinds of tissues? Where does that specificity come from?
So I'm not going to those details. There are partner proteins. Specifically what we saw is a protein called RAB18. But those things we can discuss offline. It will complicate the story more. Yeah. Okay. So 10 past 5. Great. So yeah. So this is something that was figured out by others. Sorry, I should have put in the slide. This is a figure from our paper, but essentially this sequence is found by Wang et al. In a paper about 7 years ago, where basically what they were able to show that when the kinesin motor binds to a cargo like this, and this is interesting, your work, that a part of the kinesin motor called kinesin tail domain also can bind, perhaps we think with lesser affinity, and I will come to that in a minute, to bilayer membranes. Okay, I was talking about monolayer membranes now. It binds with lower affinity to bilayer membranes, but really interesting that this binding to cargo by bilayer membranes is extremely important for the secretion of matrix metalloproteases, okay? Because phosphoric acid apparently controls that well. So we have been thinking about that. Can you block this to control the secretion of matrix metalloproteases so that's a separate issue. But anyway, so this is so these are amino acid, RIK, these are different amino acids. And this part, 901, this small, roughly 10 amino acid long part of the kinesin tail domain, this is responsible for binding to phosphatidic acid. What am I talking about? I'm talking about this binding to phosphatidic acid, okay? And that happens because there are charged groups here, okay? These are charged amino acids, and when you mutate them to make them neutral, then it doesn't bind, okay? So then the question arises that, can you now think, this is something which we are looking into, we have not done, so let me be very honest about it. Can you block this process or tone it down? Can you tone down this attachment or tone it up, depending on what you want to do, attachment of kinesin to phosphatidic acid, to regulate the outflow of fat from the liver, okay? Now, to begin with, this would seem like a stupid thing to do, okay? Because kinesin does so many other things inside cells, okay? If I am going to, play around with the attachment of kinesin to a vesicle, then it will create havoc, okay? But apparently not, okay? So that is the interesting part, and that's what I wanted to show you here. Oh, not here, here. Okay, so this might take a little bit of time to understand, but these are hepatoma cells, so think of them as some kind of hepatocyte growing on a cover slip, okay? The blue is those fatty particles, lipid droplets, okay? The red is another kind of organelle, which has a bilayer phospholipid membrane around it, okay? So we are now trying to see the difference between a monolayer phospholipid membrane and a bilayer phospholipid membrane. Why? Because can we specifically target the binding of kinesin to a bilayer and not to a monolayer, or vice versa, okay? So what you see here, you remember that the kinesin attaches to that lipid droplet by something called kinesin tail domain. So if I make lots of kinesin tail domains in bacteria, so let's say I am kinesin and I am attaching to the lipid droplet with this kinesin tail domain, I make only this part. I cut out the rest of this, okay, and push it into cells. Then all these small kinesin tail domains should go and block the binding of the entire motor to the organelle, right? Just by law of mass action, okay? But because they are only small part of the protein, it's only the tail domain, it will not work like a motor, okay, because it doesn't have the rest of it. So I may be able to remove the motor from its cargo if I push in a lot of these small parts of the motor, right? So when we do that, kinesin tail domain, you can see, just compare these two, you will see that there is hardly any change in the pattern of red organelles, which is the lysosomes. Remember, they have a bilayer membrane. But when I do the same thing for, if I look at the blue parts, they are scattered all over the cells. They are not at the periphery of these cells, right? Which means that the blocking of kinesin is happening largely on these blue things. It's not happening on the red things, okay? And you see the same thing for endosomes. Endosomes also have bilayer membranes. Lipid droplets have monolayer membranes. You can see here that when I add here, the lipid droplets are at the periphery, which is what I had told you. Because why? Because the smooth endoplasmic reticulum is at the periphery of these cells. We have shown that. Not shown in this picture, but we have shown that. So all the lipid droplets are sitting here and perhaps they are secreting VLDL out of these cells. But when I add this inhibitor of kinesin, which blocks its binding, then I see that the lipid droplets are scattered all over. Yes, makes sense because the kinesin is not working on those cargoes. But nothing seems to be happening to the red guys, okay? Means that the kinesin is doing its job. So perhaps the binding of kinesin to a lipid membrane is different for a monolayer versus a bilayer, okay? And that is quite possible because, as I said, the monolayer is fundamentally different from the bilayer in terms of, and that's something which I will come to. Further, what we saw, if this is true, that suppose in this situation, if I am not allowing these lipid droplets to stay at the periphery and interact with the smooth endoplasmic reticulum, then these cells should secrete less fat, right? Because you're not reaching the smooth endoplasmic reticulum, because you are preventing this guy from reaching here, so this should not happen, okay? So these cells should secrete less fat, and this is something actually we showed, okay? This we showed in cells. Yeah, so there are different in vitro. I don't want to go into all of the details right now. There's a wild type situation where, so you are measuring how much fat is secreted from cells into blood. We are right now doing this experiment in an animal, okay? So what we have done is that we have cloned this kinesin tail domain into an adenoviral vector, and we are in the process of infecting the animals and seeing if you can reduce the secretion of very low density lipoprotein by blocking this pathway. That work is ongoing. So why might this be useful? I mean, it may not be useful at all. We are just doing it for our curiosity, but can we help patients who have too much serum triglycerides, okay? And that is something more and more common in a younger population these days, okay? Cardiac failure. So the primary reason for premature cardiac failure is now well accepted to be high levels of serum triglyceride, VLDL particles secreted by the liver into your blood, okay? And there are a lot of therapies people are thinking about how to treat this. The other way might be, just like too much serum triglyceride in your blood, fatty liver is an increasing problem in the younger population now, okay? In fact, I know people, friends of my age and younger, who are now have fatty liver. Can you upregulate this process, okay? and to secrete out more fat from the liver and to basically clear the fat. Kind of a naive way of thinking, it may lead to other problems, but these are things which can be tested. Sometimes they are surprising. Most likely it will not work, but one can test this, okay? All right.
Have you checked for with the inhibitor of kinesin and along with the inhibitor of PLD, and what are the conditions of trackless times in the cinema?
Yes, we have checked those things. We have checked those in animals. So if we have done a knockdown of kinesin, liver-specific knockdown of kinesin in animals, largely liver-specific, and seen that these things also work. This is published work, so I'm not going through all the details. Right. Okay. So where are we? Yeah. So basically, what I was trying to tell you is that perhaps in generic bilayer cargos there are certain ways of attaching the kinesin motor to these cargos, which I mean, there are different ways. This is not a comprehensive description, but one of the ways is that something called the kinesin light chain, which is different from the kinesin tail domain that I've been talking about until now, attaches to this membrane, and the kinesin tail domain may also attach. But if I am not a bilayer, then things may be different. Okay, then this may dominate. Okay, so we wanted to test this. Is this really true? Okay, so what we have been doing now in the lab, this is so what we can, one can test this in different ways. We are doing it. I'll show you one of the things is that, so you make this kinesin tail domain, this part, okay, in bacteria, and you take a bead, okay, it's basically a plastic bead which is hydrophilic, okay, which has charged groups on its surface, and you can coat it with a lipid membrane now, and you will form something called a supported lipid bilayer. Okay, why is it a supported lipid bilayer? Because this lipid bilayer is supported by this solid substrate inside it. Okay. However, if I now play a trick that instead of taking a hydrophilic bead with charged groups on the surface, I take a hydrophobic bead, okay, of the same size. Imagine that to be the core of the lipid droplet. It's hydrophobic now. Then I should form a lipid monolayer. Okay, so I can make these two kinds of structures, and that's what we have done. Okay, and let me not get into this because I'm running out of time. Okay, so this is the bilayer and this is the monolayer. Okay, and you can. So what we have done is that oops, I have made a mistake here. Okay, this labeling is different. Sorry about this. So what you see is that so this is the monolayer, this is the bilayer. The labeling is different. That the kinesin tail domain is shown in green here. Okay, we have labeled it with a fluorophore. That's not important. It attaches with much higher affinity to the monolayer compared to the bilayer. Okay, you can see much more fluorescence here. Okay, we have done various other things that can. So if it attaches with much higher intensity to the monolayer, then it can. If I provide enough of it, it may be able to remove the kinesin from the monolayer also. Okay, and that's actually what we think is going on. That you may have the ability to specifically control kinesin recruitment to a monolayer and not do screw up too much with the bilayers. So you might be able to control this process. But these are things that we are trying right now. We will see how it goes. So the next question I will take, should I stop here or can I have time till 5:30? What is the time? Oh, 6 o'clock. Okay. No, it's okay. I can continue till 6 o'clock. Okay, yeah, because 4:30 I started, right? Yeah, okay. All right. I will entertain you till 6 o'clock. Don't worry. Yeah. So now, so we have looked at some aspects of this. Okay. Now, let us try to think about the terminal stage of this problem. Okay. What is the terminal stage? Not really the terminal stage, but whatever. Sorry. Yeah. So this is the same picture which I was showing. So you started off, let's say, somewhere from inside this hepatocyte, and you walked along the microtubules, and you reached this smooth endoplasmic reticulum. Okay. And now, if I zoom into here, this is perhaps what is happening. Okay. Here is a lipid droplet which has freshly arrived at this smooth endoplasmic reticulum. Remember that it is reactive. okay, because I had taken, not me, it's because of that ARF1, it had less coverage of phospholipids. That is one. And two, this lipid droplet had phosphatidic acid in the membrane. Okay, why? Because there was phospholipase D recruited to it. Okay. Now, if this guy has to fuse with another membrane which is largely flat, let us assume, okay, you can't just go and fuse like that. Okay, it will help if you have phosphatidic acid on this membrane, because then you can generate these kind of neck-like structures because it has negative curvature. So remember, because it has gone there here at that neck, it is played like this, right? You will be able to generate these kind of structures. Okay, so and that's what we have. I mean, this is of course an exaggeration. So, but just to show you schematically that it is likely that this phosphatidic acid reduces the energy barrier towards this fusion or interaction, whatever you want to call it. Okay, and once that happens, there are enzymes on this membrane which can now pass on to this membrane. What could those enzymes be? Those enzymes could be lipases, which chew up lipids, okay? So they can chew up the lipids here, and what is known is that once you chew up the lipid, you can make a species, let's say like diacylglycerol, okay, which has higher permeability to this membrane. And I'm not going to the details. When it goes back into this membrane, it is again reesterified to make triacylglycerol. That is known in the context of lipoprotein. And then onwards, it is going to be secreted out from here. In response to your question, I think RAB18 is a particular protein which we think is extremely important in this whole business, because we see its levels dramatically changing on the ER in response to fed-fasted states. We are working on this. I will come to some of those results. But the problem now is, so these kind of structures are very, lot of people are trying to understand these kind of structures where one compartment inside a cell fuses with another compartment, okay? Because that is the beginning of chemistry now. Because you have one kind of content here, you have another kind of content here. Only when you bring them together, you can have downstream chemical, biological, whatever you want to call it, reactions. But how do you do that, okay? And so these kind of regions, here I'm showing it in the context of lipid droplets and endoplasmic reticulum. These happen across in many other combinations, right? Lipid droplet, mitochondria, ER mitochondria, you name it, it's there. So, but these are extremely difficult to study inside cells, okay? There are a lot of very, advanced imaging techniques that are being used, some which we are also trying, but these are extremely difficult to study inside cells, because the endoplasmic reticulum is a complicated thing. Its structure, it's extensive everywhere, and it interacts with everything, and it is not straight, it is curved. And to understand that geometry, you require, very high-end instruments, ion beam milling, all that. We don't have the capability to do that, okay? So, yes, please. It's not necessary. There may be other ways, but it helps, is all what I'm saying. Yeah. Yeah, so what was I saying? Right, so how do you study these membrane contact sites, okay? People have been looking at this by very advanced techniques. So we thought of a poor man's way of doing it. We never thought it would succeed, but here is what we tried, okay? What we thought is that let us try to make this complicated endoplasmic reticulum into a two-dimensional structure, okay? So the purification of endoplasmic reticulum from the liver of rat is very well established, okay? There are protocols to do it. And so if you take the liver and the liver tissue, you will get lots of cells there, right? And so different cells have different contents of proteins, lipids, other constituents. And if you take that liver mush and put it into a tube with different sucrose density gradients, and you spin it, different components come at different places, okay, depending on their densities, okay? And you can make a fairly pure fraction of the endoplasmic reticulum. This endoplasmic reticulum, now that you have extracted from the liver, is not that kind of a complicated structure anymore, but rather it has broken up. Obviously, right? You have taken it to this procedure, it has broken up, and you have small, small vesicular structures. Okay, these are called microsomes. So these microsomes are what you get outside the cell of that complicated structure in small vesicles. Okay, 100 nanometer, 50 nanometer vesicles. What we did, so these are those microsomes. Okay, do you understand? This is some mimic of the ER. It's not exactly the ER, obviously, because I've done a lot of things. We simply took these microsomes and we took a cover slip which we made hydrophilic, okay, by plasma cleaning, and we simply flow in these microsomes onto this, okay. And you can see, okay, so here is a red color which stains the lipids, okay. And we see that you can actually you flow it into this chamber, all these little vesicles, and you form a beautiful bilayer membrane on this, okay. So this is a sort of a mimic of the endoplasmic reticulum. Of course, it is not exactly the way it is inside cells, but this is the best we could do, okay. Now, so imagine on this cover slip, I have this endoplasmic reticulum mimic sitting on this, and we have verified, I'm not going to go into all the details here, we have verified that many of the lipids and the proteins which are normally present in the endoplasmic reticulum are also present on this bilayer membrane, okay. So now, if the lipid droplets inside those hepatocytes were going and interacting with the endoplasmic reticulum, can we test that in this assay? Okay, so simple, you have this membrane on the cover slip, you purify lipid droplets from the liver, okay, and you flow it in and you leave it for some time, let them interact, then wash them off. If there are stable contacts, post washing a larger number will survive, right? If they are not stable, when you wash, they will all go away, okay. So now remember, the claim was that in the fed state, these lipid droplets interact with the ER. That was the cartoon. In the fed state, this interaction happens. In the fasted state, kinesin is disabled on this because you don't have insulin, so this doesn't happen, okay. Now, this is again a poor man's way of doing it, but now so this is the experiment where you take a certain number of lipid droplets, you put them in. Before you wash, you see so many lipid droplets, and after you wash, you see so many lipid droplets. Okay, you simply count the ratio. Just find the ratio, and I'm sure I've forgotten the slide. Yeah, no, I haven't. Okay. Yeah, okay. So, sorry, I forgot the bar graphs, but when you take a combination of fed lipid, so what can you do? You can prepare lipid droplets from the liver of a fed rat, you can prepare endoplasmic reticulum from the liver of a fed rat, or you can do the same thing from a fasted rat. So you have 4 combinations, right? So when you take a fed-fed combination, you see that practically all the lipid droplets are sticking, okay? But when you do it with a fasted combination, you see hardly any lipid droplets remaining, okay? Now, I'm not going to go into the details of all this, we can discuss it. What we could also do is that we could, maybe I have that actually. We could do another kind of assay. Yeah, here. No, not here. Yeah, so we could do another kind of assay where we hold a single lipid droplet. So this is a lipid droplet from a fed rat. Underneath it is this endoplasmic reticulum membrane from a fed rat. Okay, I simply shine a laser on this lipid droplet and trap it in an optical trap. Okay, I trap it in an optical trap like this, and I move this membrane underneath and see if it is binding or not. I can measure the forces, I can measure the binding energy of this, okay? So we have done that, and what you see is that when you have a fed combination, then nothing happens now. Yeah, you can see that the underlying membrane is moving, and this guy got stuck to this membrane and moved out of the optical trap, okay? When I do the same experiment with a fasted combination, that does not happen. I mean, it's moving in the other direction, but doesn't matter. You know, so it doesn't stick. There are very weak sticking events, and we can calibrate the optical trap for this lipid droplet, and we can measure the approximate force, and we believe that when this gets bound, it is bound with a force greater than 20 piconewtons. Okay, but that's something which I mean may not be so important for this, but right. So now there are very interesting questions I believe one can try to address here, because this binding energy of this lipid droplet to this kind of, you know, so remember that this is bound to this bilayer membrane underneath. The binding energy is proportional to the square of the line tension by the lipid droplet surface. So the line tension is basically the energy along the interface in a line, in a curved line, and the surface tension is the energy on the surface. If you want to increase the surface by so much, how much energy do you require? Okay. And why is the line tension different from the surface tension? Because the lipids at the interface may be different when you're not at an interface, right? So that's why it's different. So, right, so there are interesting questions, but what I want, you know, the way we look at this problem is something like this, okay? So, it is very well known that there are extensive interactions between the endoplasmic reticulum membrane and the lipid droplets. The lipid droplets are born at the endoplasmic reticulum. They, you know, when you feed cells with fat, that's when the adipose fat is coming to hepatocytes, the fat actually emerges out from the lipid droplets and goes into the cytosol to make the lipid droplets. So imagine this is the endoplasmic reticulum, the mother duck, and all these little ducklings, the lipid droplets go out into the cytosol and they interact with all kinds of organelles, okay? And then at some point they have to come back to the ER, because at least in the hepatocytes where they have to quote unquote die, okay? Because that is where you will catabolize these lipids again and send it out. So these guys go out to party every day and then, you know, come back and then they are, you know, recycled. So this is happening in the liver, and we feel that we have been able to understand at least some of the aspects of this and try to understand what happens at the level of physiology when you try to connect it with the molecular level processes. 5:30. So, okay, I think I will stop here. And do I have anything more? Yeah, I have. I would like to, you know, so this is IIT-Bombay, really beautiful from the top of the hill in the monsoon. You can see the Powai Lake here, place I frequent quite a lot. And these are people from the lab. And the last part where we made these bilayer membranes is done by Sukrut and Jagjit with help from others in the lab. Shubham is working on this monolayer versus bilayer. Dvitiya, who is here also, is working on other aspects which are more biophysical, which I will come to maybe tomorrow or day after. That's it.